this very important invitation. This is my first time here in Gothenburg. Very beautiful city, very warm people. So I feel very good and comfortable here. I would like to try to talk about the main activities that we are working in my institute in Chile. And the topic is the pesticide use in the agriculture. And this is the outline of my presentation. I would like to talk a little bit of my institute, INIA, this is the name. My research group, our laboratory, the research lines, and some conclusions about the research. INIA is an institution that it's part of the different institutions related with the productivity in Chile. As you know, Chile is a country, very long country, that we have different conditions, very interesting conditions. You can see here, from the north to the south, we have one of the driest deserts in the north. We have all the Pacific coast, and in the south we have Patagonia. So we have different climate, different landscapes through all the country. And of course, the geographical conditions are exceptional for the productivity. And of course, there are some special geographical areas to produce different crops and of course different type of food. Here you can see Chile is it's the longest country in the world. So here you can see we have our neighbors, Bolivia, Peru, here Argentina. So we have natural boundaries here. And the agriculture, it's a very important activity in our nation. Here you can see we can separate our country in different areas. In the north of Chile, you can see the special agriculture is related to horticulture. And the little north here, we have pisco production, is a kind of drink that we produce with the grapes. And of course, we have horticulture production and goat raising. In the central area of Chile, here in the valley, maybe you know the Chilean wine. All the Chilean production, the Chilean wine production is here. The mainly zone is in the valley of Chile. And of course, we have fruit exportation from this zone. And through the country, going to the south, we have the frontier, and that we have the cereal production, the livestock, and forestry. And of course, we are looking for opportunities here in the Atlantics. So we have a very different agricultural activities through the country. So what is INIA? What is this institute? It's the main institution of Chile of agriculture. Belongs to the Ministry of Agriculture of Chile. And it's a non-profit organization. Our budget depends on the Ministry of the Agriculture, and leads to the knowledge and technology development in the agriculture. And of course, we have many challenges for the farmers, for the national production and the exporters in Chile. Of course, we know that we need to look for solutions and to look for innovations, and of course to improve the competitiveness of the national the agricultural sector. INIA is the presence is from the north to the south with 10 regional centers from the north to the south. We have technical office, experimental centers, gene banks, laboratories, and libraries. These are the main research programs that we have in our institution. You can see we have very different areas because we have very different geographical conditions. So we have crop research, horticulture production, fruit production, livestock systems, genetic resources, sustainability and environment, food research, technology transfer and extension. So we try to coordinate all these research lines and try to provide to the growers in Chile all the solutions related with different areas in the agriculture.
Sorry, do you mind just keeping yourself a bit more further closer to them? Otherwise, they won't see you.
Okay, sorry.
You're supposed to be in the picture.
Okay.
So move a bit forward, right?
Where?
No, I mean, a bit closer, like that.
For the camera. Okay. Okay. In our institution, we have some strategic research. It's like okay. Yeah. The main areas are related with the climate change, related with the water use efficient, and of course the genetic resources and plant breeding. The food security is one of the main areas now in Chile. Chile is a country that we produce a lot of copper, and we try to improve the food production. It means that we need to put more effort about the food security. So our purpose is to reduce the amount of pesticide in the agriculture. We need to evaluate the dynamics, the behavior, and of course to evaluate the risk analysis of the use of different agrochemicals, and looks for solution with plant breeding programs. And of course the sustainability is very, very important. The land management and the use of organic waste, it's a very important topic. And of course all these areas are very important because the research will be necessary to transfer to the farmers in Chile. This is the purpose, to look for solutions in our labs and then go to the farms and try to teach the farmers how they can improve their activities. So we have a very important interaction with the farmers. So we are listening what they are talking about problems, talking about how they can improve the agriculture. And of course, in this relationship, we try to look for the better solution for them. Well, here you have South America. We have here Chile. Okay, Santiago is the capital. Here it's in the middle of the country. And here is one of the center, La Platina. This is my research center in Santiago. This is our group. We have different people working in the lab in different areas, all related with the analytical chemistry. And this is our web page if you are interested in about what we're doing specifically. Our lab, I'm responsible for the lab of pesticide residue in Chile since 2017. And one of the main focus of our work is to obtain the accreditation, the ISO 17025. This is a very important standard to have very important quality of results. So for this reason, 1 year ago, we put a lot of effort with our team to try to obtain this certification. So we approved the process in the last month. So we will have the next month the certification that we have this ISO that is very important because we are working with pesticide. So we need to prove that the quality of our results are very important. So it means we always we try to look for proficiency tests, trying to publish with the result of our analytical data. And of course, this step for us, it's very important. So of course, because when you work with pesticide, it's very important the image of your lab, how you can demonstrate that you have all the skills in your lab, and then the customers or the researchers are totally relaxed about the result. Okay, this is a hard process. During 1 year, we were working about all the traceability, the validation of different methodologies, and of course, it's mean calibration tests, different organization of the people on the work for their activities. Looking for this, the accuracy. Of course, all the method that we have needs to be validated with different indicators. One of the most important methods that we are working in the lab, it's the determination of pesticide residue in vegetables and fruits. So it means this is part of the accreditation. Of course, we work with soil, with water, but this is part of the accreditation. And of course, when you are working with the ISO, it's very important to have traceability, because if you are doing something wrong, you need to immediately check where is the problem in the lab. So we have a lot of papers in the lab, checking every step to see how we can improve all these steps, like using corrective actions, preventive actions, and of course the control of the records. It's very important. Okay, the research lines in our group, as I mentioned, the agriculture is very, very important in our country, and of course it's an important source and for the economy. You can see here the fruit exportation represents the 40% of the agricultural GDP, and the country is one of the world's leading exporter on fresh fruit and wine. So it's a big challenge, because all the farmers in Chile are always asking how they can improve their productivity. And of course, we know around the world all the regulations are very restrictive. For this reason, we need to look in each country that we want to export, how we can improve to don't have any problem about the process and the exportation. Of course, in the agriculture, it's necessary the use of pesticide when you need a high demand of food around the world. Our country wants to be a leader in 10 years more about food production. Not only about fruit, we want to export raw materials for nutraceuticals or for different purpose. But one of the issues is the use of pesticide, because we know that is a very difficult topic to fix. So you have here some information about the pesticide sales and pesticide use in Chile. Unfortunately, we are increasing the pesticide use in our country. You see the use of pesticide increases faster than the total agricultural production and the expansion of the agricultural land. One factor is the climate change is changing all the conditions in the agriculture. So for this reason, we need to see how the behavior of the pesticide in the environment is changing too. You can see here, Chile had the highest average annual increase on pesticide sales between OECD members countries. And since 2000, the increase of the use of pesticide is very, very important in Chile. Some studies are published and indicate that Chilean farmers, they spread a lot of pesticide and often more than is necessary. So it means if you have a pesticide program that it will be necessary 10 or 11 applications, Chilean farmers are using 15, 17, and they don't understand what is the real impact in the environment and the human health. And of course, there is a risk because of the use of pesticide in soil and water. And at the moment in our institute, we need to look how we can contribute for the decision process of the government to reduce or to remove some pesticide from the market. So our purpose is to provide scientific information and of course to see the impact of this pesticide residue. And we are looking for different strategies because in Chile we don't have restriction about pesticide sales. So everybody can go to the market and obtain pesticide without any condition. So for this reason, we are working with with Jessica to see how we can put some strategies in Chile to reduce the use of pesticide. And of course, we are always with the Chilean producer in a big discussion because they need to produce, but we need to protect them. So to keep the balance, it's very difficult. So we are in the last year, we were in big discussion about how this balance can be really effective to reduce the contamination in the environment, of course, and the impact of human health. I want to show you some information that about some activities in our group related to the pesticide residue in soil and water and some green technologies that we are using to decrease the contamination. Here, this is a very common practice in the farms. You can see here how the people are using and handling the pesticide when they are loading, filling, mixing, or just washing the machinery. So in all these cases here, you have different concentration of pesticide. In some cases that you have very small quantities with high concentrations or large quantities with lower concentrations. But depends on the chemical properties of pesticide, you have more fast transport or more fast transformations. So it means we have a source of contamination. So it means how we can decrease this. 30 years ago, in the University of Uppsala, here in Sweden, they create a technology to reduce this contamination in water. In the last year, we have many collaboration with Maria del Pilar Castillo and John Stentrum from Uppsala University and JTI. And we were collaborating about this system to reduce the amount of pesticide in water. In Chile, we have some pilot biopurification system that is very simple. It's an organic biomixture, and you have here soil. In this case, it's a typical Chilean soil with some organic matter, different physical-chemical characteristics, and we have peat and, of course, straw. So this biomixture we put in this system, and of course we are evaluating high concentration of pesticide, and we are checking how is the impact in the microbial activity in soil. And here we have some experience about to reduce some high concentration of important pesticides that we found usually in Chile, like atrazine, like ipredone or chlorpyrifos. And of course, using this kind of biopurification system, we can reduce a very, very high concentration, and 40 days that we can reduce this high concentration for this pesticide. We are trying to demonstrate that this technology can be used for the Chilean growers. So the next step is to go to the Ministry of Agriculture and to present the information that this technology is a very cheap technology to reduce the contamination of pesticide in water. We saw that the impact in this biomixture at the beginning, with high concentrations, we have some impacts in bacteria, fungi, and actinobacteria. And through the time, we saw that the capacity of this biomixture can improve the population of these microorganisms. So it's very important because most of these microorganisms, they produce extracellular enzymes that are involved in the process of biodegradation of the pesticide. So we saw that the influence of this pesticide and the microorganisms are not very important to see if they can live in this biopurification system. And we saw some enzymatic activities that are related with the process of biodegradation. And of course, we saw too that the activities are not very influenced in for the presence of the pesticide. Another very important topic that we work in our lab is to identify which kind of pesticide residue are very important in the agriculture. During 2 years, we took 116 samples from the north to the south here in Chile, in 3 regions, the main regions of Chile that they produce a lot of vegetables and fruits. So we decided to go to take the samples, and of course we have 2 realities in Chile. The reality of the exporters, that they are very, very, very worried about this, and the Chilean farmers that produce for the national consumption. 2 realities very, very different, because most of the exporters in Chile are very worried about how pesticides are in the fruits. So for this reason, in fruits, usually we don't look for pesticide residue. We have from the national authorities some monitoring programs, but in vegetable production for the national consumption, it's very complicated scenario. We took the samples of leafy vegetables, lettuce, spinach, and chard, and we took the samples to the lab. We made the evaluations, analytical evaluations, and we make human health risk assessment. We determine different parameters, and we evaluate with the scenario of the World Health Organization, and they describe that the food consumption of leafy vegetables in Chile is 2 grams per day. So for this evaluation, we consider this scenario. And we made the method performance and quantification. These are the main pesticides that are detected in leafy vegetables. And you can see from the total, 27% of the sample were over the MRLs, the maximum residual levels of the Chilean regulation. So this is a big problem. And most of the problems coming from lettuce and chard samples. You can see here we found different pesticides like boscalid, chlorpyrifos, lambda cyhalothrin, methamidophos, and the frequency you can see here more than 10 times we found in the samples. Here you have the concentration, and of course this is a very problem. You can see, for example, methamidophos, we found sample like 29 milligrams per kilogram, and the MRLs it's 0.001. So this is a very big problem, because of course we know that this pesticide is very toxic. Most of the countries are removing methamidophos around the world, and one of the purposes of our lab is to show the evidence that this pesticide needs to be removed as soon as possible from the Chilean market. But we need to show the evidence, and one of these activities is to provide the information about the concentration that we found, and we use the risk assessment to see how the impact in long terms will be affecting to the Chilean population. Here you can see when the estimated daily intake, we found that cypermethrin, methamidophos, and mancozeb mainly were the main pesticides that are producing important problem. One of the problems in Chile is that we don't have enough information to take decisions. You can see here, this is a report from the government that was published in December of 2017. We saw that in this report that the total samples in 2015 in Chile were analyzed, almost 2,000 samples, and most of them were fruits and vegetables too. This report showed that the Chilean regulation of maximum residual levels of food, of course based in the Codex Alimentario, they found that 170 samples were with problem with pesticide residue, and this is an official notification in Chile. And of course most of the problems come in the vegetable production, and they found that the main pesticides are methamidophos, chlorothalonil, and chlorpyrifos. In our study we found similar results. They they didn't publish the concentration of each pesticide. They only published that the amount of pesticide that exceed the MRLs. For this reason, it's difficult to take decisions if you don't have all the information. And of course, we know that this is a very important topic. So we decided to evaluate the pesticide residue and fresh, ready-to-eat vegetables. And this is a very complicated problem because we took samples from the schools that are supported from the government. And of course, HUNAEF is the National Board of Schools, Aid, and Scholarships. And we found in Metropolitana region in Santiago, in this school, we found very interesting data to show the evidence that pesticides are it's a very hot problem in Chile. We took the samples to the lab, we made the validations, and we make the human health risk assessment. But in this time, we took 3 scenarios. The scenario of the World Health Organization, a national survey of Chile that was published by the Ministry of Health, and the official data from the government that coming from the Ministry of Health. So some results, we found that in lettuce, we don't have problem about pesticide. In the last experiment that we found important concentrations, we measure from the vegetables in farm. And the total samples here, we have more like 10% of contaminated samples. Here, in this data, you can see here the 3 scenarios: the World Health Organization, the national survey, and the official data from the government. And here you can see the consumption. For this evaluation is 2 grams per day, and the Chilean national survey is 35 grams per day, and the Ministry of Health, the official data is 67 grams per day. So when you do this evaluation, you can see the ratio between the estimated daily intake and the admissible daily intake. And you can see here, for example, methamidophos is 66 times the concentration that you can eat in one day. So it's very difficult to explain how this is possible. Okay, this is the reality of vegetal production. We have chlorpyrifos, methamidophos, and lambda celotrin that are the main pesticides that are producing these problems. And here, when we estimate the chronic hazard index, here, if you have an estimation that it's over 100, you will have a long-term problems about the pesticide residue that we found. So in the case of the scenario of the official data of Chile, for men and women in this age group, you will see we have problems with the concentration that we found. And the average is the same scenario. For all the age group, we have different problems about long-term exposition of these pesticide concentrations. Some conclusions. In our research group, we are working with sustainable technologies to try to reduce the amount of pesticide and, of course, the contamination. and the biopurification system will be a very important alternative for the farmers in Chile. In the case of the Chilean population that are consuming vegetables, it's exposed to health risk because of the concentration of 3 main pesticides that we found. In this case, the main pesticide is methamidophos. And of course, our main objective now is to go to the farmers and of course try to evaluate how we can remove the methamidophos and the agricultural practices, and of course trying to see which alternatives are better to control the pest and disease, of course. Okay, thank you very much for your attention.
Thank you very much. Let's open up the floor for questions and discussion. Thomas.
Just a question, clarification-wise. How do you set the MRLs? So what kind of methodology do you use for that? Is that elementarius, or where do they come from?
In Chile, the MRLs are based on the codex,
Okay.
are the same of the codex. And if you don't have some MRLs and codex, we use the European Community or from United States, but are based on different countries.